Okay, so today we're going to talk about Chapter 4. We briefly discussed what a cell is. Remember in Chapter 1, I referenced the difference between a prokaryotic cell and a eukaryotic cell. Today we're going to talk about them more specifically, the defining structures, and then the functions of each one of these internal structures called organelles. To start with, let's look at what we call the cell theory. So there's 3 statements in the cell theory. The first one says that all organisms are made up of cells, right? Remember what an organism is, back from Chapter 1. The second statement says the cell is the basic living unit of organization for all organisms. So remember, out of those 7 characteristics that define life in Chapter 1, the smallest living unit is a cell. So atoms, molecules, elements, carbs, compounds, they are not living. They're just molecules that make up cells. And the third statement says that all cells come from pre-existing cells. So this may seem like common sense to us, but remember back in the day, guys, we didn't know this. They didn't know what DNA was, or mitosis, or meiosis. So what this means is that cells just don't spontaneously appear in random places, right? That they must come from an existing cell. There are 2 types of cells, the prokaryotic cell and the eukaryotic cell. Remember in Chapter 1, I referenced that pro means pre, karyo means nucleus, right, or a sac, versus eukaryote, so EU means true, karyote means nucleus. And the main difference between these 2 types of cells is the location of the genetic material. So in a prokaryotic cell, the DNA is found in this nucleoid region. Notice it's a region, right? It's not a sac. Without a membrane separating it from the rest of the cell. Whereas in a eukaryotic cell, chromosomes, which is your DNA, genetic material, are found in the nucleus, which is a membrane-bound enclosed organelle. And so in the next slide here, you can see, of course, now these aren't relative to size. On this particular slide, it looks like they're the exact same size, but they're absolutely not. Remember, prokaryotic cells are much, much smaller, back from Chapter 1. I just blew it up so you can see the differences. You'll notice, first of all, the major difference here, again, here's that nucleoid region. Now, this isn't a sac, it's just a region. It's an area where the genetic material, so the DNA and RNA, or sorry, the DNA or RNA could be found. Within the eukaryotic cell, this is the nucleus. So it's definitely defined by a membrane, and it actually houses the genetic material, or the DNA and or RNA. You'll also notice one of the major differences here is that in the eukaryotic cell, we have these internal membranes. So if you look here, here's your mitochondria, Golgi apparatus. Here's a bunch, looks like a bunch of channels down here. Here's some little vacuoles and sacs. And so these membrane-bound organelles are what define eukaryotic cells as well as being different from prokaryotic cell. Inside of the prokaryotic cell, as you look here, I mean, you can see little things here and there. There's a few ribosomes, but it definitely lacks the organization and ultimately these organelles, which are like the tiny organs of the cell, compared to the eukaryotic cell. Now, why this is important is because, if you remember, prokaryotic cells are your bacteria cells, right? They're very tiny, they're small, they're your E. coli, your staph, your MRSA. Eukaryotic cells are your plant and animal cells, and they're obviously much more complex. They can carry out much more complex functions. And the reason being is because when you membrane bound these organelles in here, it allows this particular cell, or the eukaryotic cell, to carry out specialized functions, right? Because it's separate from all the other things in the cell. Whereas in the prokaryotic cell, all of their metabolic processes and functions must occur in this mass chaos of just space, right? So they'll never be complex. Eukaryotic cells are ultimately able to carry out these functions because they have these specialized organelles which carry out specialized functions. And we'll talk about what these functions are in just a minute. Basic structure of your eukaryotic cell. So all eukaryotic cells contain a cell membrane, right, which is that membrane that surrounds the cell, protects it. They all have what we call a cytosol. So the cytosol is a semi-fluid substance within the membrane. So it's kind of the jelly-like substance that all of the organelles float around in. They all have also known as a cytoplasm. So the cytoplasm is just the cytosol plus the organelle. So it's the totality of the cell. And all eukaryotic cells have what we call chromatin. And so chromatin contains the chromosomes, which have genes in the form of DNA. So this is your actual genetic material that's found in the nucleus. If we take a little closer look here at that cell membrane that surrounds the eukaryotic cell, you can see, if you look down here, guys, at the very bottom, what do you notice? Those are our phospholipids that we just talked about in chapter 3. And remember, those phospholipids have these polar heads, so these little gray balls right here, and they have these non-polar side chains. So you'll notice that the cell membrane is made up of this bilayer, or this double layer, which creates this internal hydrophobic region, which means that it's going to be quite difficult for substances to get across the cell membrane, so to get into and out of the cell without help. And these big purple blobs right here, these are called proteins or membrane-bound proteins. And it's these proteins that are going to help things get in and out of the cell. So it's going to help the cell take in nutrients like big carbohydrates, and it's going to help the cell to expel nutrients or expel waste. The cell membrane functions as a selective barrier. So by selective, right, it means that it selects what comes in and out. It allows the passage of oxygen, nutrients, and ultimately waste. And so this bilayer right here of phospholipids, which makes up the cell membrane in eukaryotic cells, I believe it's the next chapter. We will spend a whole chapter talking specifically about the structure of the cell membrane and how it actually functions. For today, I just want you to be able to recognize its structure, and its main purpose or its overall function is to really protect what gets in and out of the cell. Eukaryotic cells, as we said in the past, are much more complex than prokaryotic cells. Within the cytoplasm, we have these membrane-bound organelles. And so I referenced earlier that an organelle, it's just like a tiny organ, right? So just like you have organs, you have heart and you have a digestive system, which is made up of organelles like the small intestines and the stomach and the large intestines and lungs. Within the eukaryotic cell, they also have these tiny little organs which carry out specific functions. These organelles are specialized in structures and form and function, and they're much larger than prokaryotic cells. Here is a picture of a typical eukaryotic cell, and we are going to reference or go over most of these organelles. So every one of these things that are labeled on here, either organelles or basic structure. You should be able to look at this diagram of the eukaryotic cell without the labels on it and identify what the organelles are, or the structure, what the basic function is. And then ultimately be able to identify what is not found in animal cells, and on the next slide, what is not found in plant cells. So what organelles are exclusive to an animal cell and a plant cell. I'm just going to quickly right now go over the basic organelles and structures, and then we'll look at them more specifically in future slides. So the first organelle here that you should easily be able to recognize by now is the nucleus, right? It's the largest organelle in the eukaryotic cell. You can see it has this membrane on the outside. And the nucleus right here is made up of three things. So the chromatin, again, for the purposes of this course, is just another word for the DNA, right, or the genetic material. There's a nuclear envelope on the outside. And then in the middle right here, if you see this densely stained area, this is called the nucleolus. Continuing on, you'll notice there is this it looks like a bunch of channels. This is called your rough ER, or endoplasmic reticulum. Here's your smooth ER right here, which looks like the rough ER, but you'll notice it doesn't have those little dots all over it, so it's called smooth. Those dots are actually ribosomes. We then have a Golgi apparatus, which is considered to be our shipping center of the cell. We have this kidney bean looking organelle, which is called the mitochondria. This is where the energy is produced in the cell. Lysosome down here, this little purple sac, the stomach of the cell. The cytoskeleton, which is labeled in several areas. It's these little lines throughout the cell and functions just like our skeleton does, right? It helps us move, it provides support, it provides internal structure and function. There's something called microvilli, and not all eukaryotic cells have microvilli, but this one does. Peroxisome, these little centrosomes right here, these barrel-looking structures. Flagella, right, so this long whip-like structure. And then down here on the right-hand side, these four structures are not found in animal cells. So you do not see a chloroplast, you don't see a central vacuole or a tonoplast, you don't see a cell wall, and you don't see a plasmodesmata. You do see these in the next slide, which is your plant cell. So you'll notice they have a lot in common, right? The difference between the animal eukaryotic cell and the plant eukaryotic cell. So they still have mitochondria, they still have a Golgi apparatus, they still have centrosomes, they have a nucleus, they have a smooth and rough ER, they have ribosomes. What they do have that plant cells don't have is they have this thick outer cell wall right here that you can see, gives that plant cell some shape, some rigidness. They also have these green structures, which you see right here. These are chloroplasts. And they have these little holes right here within that cell wall, which are called plasmodesmata, which are going to allow the plant cells to share some substances and information with adjacent cells. The last structure you'll notice here that the plant cells have that animal cells do not is called that central vacuole. And so this central vacuole is going to be responsible mainly for water regulation and some nutrient regulation, but you did not see this in the animal cell. Down here in the bottom right-hand corner, you'll see there are also several structures here that are not in the plant cells that we did see in the previous slide of the animal cell. Plant cells do not have lysozymes, they do not have centrioles, and they do not have flagella for the most part. All right, so we just looked at these organelles, or also what we call these internal membranes. So to review, in eukaryotic cells, internal membranes allow for partitioning of cells into compartments. So we have these little special compartments, which creates different local environments within those compartments. It allows us to compartmentalize functions, and the membranes for different compartments are specific for that function. So for example, down here, this is a mitochondria organelle, and you can see it's highly folded on the inside, and that's going to be very important when we talk about creating energy within the cell. Here is an example of a chloroplast cell. Here's some Golgi apparatus right here, and here is some endoplasmic reticulum. Now, I want to point out, up until now, we've mainly just seen like cartoons of cells and structures and atoms. In this particular chapter, we start seeing some real microscope slides, right, or views. And so in your microscope lab, which you're going to perform on the next lab, you're going to use a light microscope to view some typical bacteria, plant, and animal cells. However, you won't see the internal organelles like this in this much detail. The reason you're able to see them in this picture, these are actual cells, right? They were actually viewed using a microscope, is there's different types of microscopes that are really important, particularly in cell biology. The reason that you're able to see these detailed internal structures is because we're using something called a scanning electron microscope. And so it uses electrons to actually scan through the organelles or through the cell, and you're able to see these highly detailed structures, which is pretty cool. So throughout the slide, when you see these nice black and white structures, these are actual pictures of cells. They're not cartoons, right? But you're only able to see them because of these different types of specialized electron microscopes. So they might be scanning electron microscopes, which allows you to see internal membranes or internal detail of the cell. Another type of microscope is a transmission electron microscope, which we'll see some pictures of in just a minute. All right, so let's start off with our first major organelle called the nucleus. The function of the nucleus is that it contains the genetic material, right, also known as the DNA or RNA of the cell. Most genes are in the nucleus, and some genes are located in the mitochondria, but for your purposes, we're going to focus on the nucleus. The size is about 5 microns in diameter, so again, that just means it's the largest. Of course, I have some activities up here as well that you may click on for a further or deeper explanation or just to rehear this information again. And this is a great picture of a scanning electron microscope of the nucleus of the cell. So you can clearly see, here's that nuclear envelope which surrounds the genetic material, which makes it an organelle. It's also a double membrane, just like the cell membrane. Here are the nuclear pores. You can see right here, it's very tiny. Nuclear pores are important. Why? Well, because the DNA or the RNA eventually is going to have to travel outside of the nucleus, right? So it allows a passageway for that genetic material. There are some other terms in here that you don't need to worry about, but you should know nuclear pore, the nuclear envelope, and then right here in the middle, this is a densely stained area, which is generally located towards the middle of the nucleus, and this is called the nucleolus, which is responsible for making the ribosomal RNA, which you don't know what that is yet, but we will talk about it in the future. The nuclear structure, you can see that it's separated from the cytoplasm, as we just saw, by this double membrane on the outside. It's fused with spots, which form these little pores right here, which you just saw in the previous slide, which allows large macromolecules and particles to pass through. So again, here it says what kind of molecules need to pass through. Well, we don't want to really let anything in, right? Maybe a few hormones or a few small substances, but the major things that we need to let through is we need to let out that messenger RNA so it can eventually meet up with the ribosomes in the cytoplasm to produce proteins. This structure, the reason I put this figure on here is because I wanted to show you an example here of a different type of electron microscope. So remember the previous slide, or in this slide, this was a scanning electron microscope, so you can see the internal details very well of the cell. This, on the other hand, is called, excuse me, a transition electron microscope. And you can see in the transition electron microscope, you can actually see the 3D structure much better, right? And so again, you don't have access to these microscopes, but it allows you to see the external structures. And so here's the actual surface of that nuclear envelope. Here's some close-ups of the pores, right, that let the genetic material leave the nucleus. And then this is the nuclear lamina, which is kind of analogous to the cytoplasm of the rest of the cell. This is a big picture of the nucleus's role within the cell. There's a lot of detail in here, and I know I always say this, but in future chapters, we will talk about this whole process on a molecular level. For today, I just want you to know that it's involved in something called the central dogma. And so right here, this double helix, hopefully you recognize that this is DNA, right? That DNA double helix, which is our genetic material that's housed in the nucleus. Eventually, this DNA will be transcribed within the nucleus into something called a messenger RNA, which it's decoded, this long red ribbon. The messenger RNA will then be exported out of the nuclear pore, as you can see. It will eventually bind with the ribosome, which is this large brown unit, in order to produce a polypeptide, right? So we've talked about polypeptides, also known as proteins, which are made up of amino acids. So the central dogma is going from DNA to RNA and then ultimately to a protein. This whole process right here, the transcription translation process, is talked about in future chapters. But I just wanted to show you the complex things that occur in the nucleus. And also, you know, when we say that the nucleus houses the genetic information, this is what it means. It houses the DNA and the RNA. A little more about the nuclear structure. It says within the nucleus, DNA is organized into fibrous material called chromatin. So it's the chromatin When the cell prepares to divide, this chromatin will actually coil up into these little X-like structures here, which are called chromosomes. And I showed you this because a lot of times, depending on how much experience you have in your previous biology classes, you may see these X's, right, as chromosomes, or on like a Meteosaurus, or when we're talking about karyotyping, or on the news. And it's all the same thing, guys. It's just in different forms depending on what the cell wants to do, right? So the chromatin, the chromosomes, and ultimately this is all together called DNA, or genetic material. Lots of different terms meaning the same thing. And then you'll also notice here's that densely stained area, the nucleolus in the middle. Again, here is a nice picture of a scanning electron microscope. Here's that densely stained area, the nucleolus in the middle. The function of the nucleolus, it says, is to produce ribosomal subunits for the RNA and eventually for proteins. So this is your ribosome down here, which is responsible for protein productions, and we'll look at him a little later. All right, our next structure that we're going to talk about is called the ribosome. And so the ribosome were those little bitty dots that were all over that endoplasmic reticulum that I pointed out in an earlier slide. The structure of the ribosome is it contains two subunits. So you can see here's a larger subunit, here's a smaller subunit. And in this particular electron microscope view, you can see there are actually free ribosomes, these little dots right here in the middle. And there's also bound ribosomes. So the bound ribosomes are actually stuck here to the endoplasmic reticulum, right? Therefore defining if it's rough or if it's smooth ER. The function of the ribosome is ultimately to produce proteins. So remember in chapter 3, we talked about protein as a macromolecule, right? Which is made up of those 20 amino acids. And the most important thing, I'm just reviewing here, that we said about proteins was their three-dimensional shape. Remember, it was so important that they fold properly from that primary, secondary, tertiary, and ultimately a quaternary structure if it was needed, right? But you guys know the structure and function of proteins. The structure that's responsible for creating these proteins is the ribosome. There are two types of ribosomes, as we just saw. There are free ribosomes, again, which are suspended here in the cytosol. Their function is to synthesize proteins, which function within the cytosol. And then there's also bound ribosomes, which are attached to the outside of the endoplasmic reticulum. And I know it's a little difficult to see right here. If you look really closely, you can see it. We'll see a better picture in a minute. Bound ribosomes are responsible for synthesizing proteins that are ultimately going to be exported for the membrane, right? And so two different functions if you're a free ribosome or if you're a bound ribosome. Okay, now I want to take a minute to talk about a few of the organelles collectively, known as the endomembrane system. So the endomembrane system is just what it sounds like. Endo means inside. It's a membrane system. So you can see down here, as things leave the nucleus, they take different pathways, right? So all these different pathways will play key roles in whether we synthesize or break down macromolecule within the cell. And so here are the major players down here. We have the smooth and the rough ER, the Golgi apparatus, some vesicles, some vacuoles, and eventually we may also go to the lysosome. So the endomembrane system is the system that we use to modify particular proteins or macromolecules within the cell. We'll look at each one of these players in the endomembrane system, starting with the endoplasmic reticulum. So you can see it up here. It's this large, looks like this channel-like system, which actually is attached in certain areas to the nucleus. So the function of the ER is to manufacture proteins and perform many biosynthesis functions. So remember, it's manufacturing proteins, but if you look here on the ER, you see those little dots right there? Those are the ribosomes. We already said who makes the proteins? The ribosomes, right? So this is where protein synthesis occurs on the ribosomes which are bound to the endoplasmic reticulum. The structure is a membrane which is connected to the nuclear envelope and extends throughout the cell, for much of the cell, accounts for about 50% of the membrane. And there are two types, the rough ER, which has bound ribosomes, and the smooth ER, or endoplasmic reticulum, which does not have any ribosomes. Here you can see, I just wanted to show you an electron microscope view. So here's our cartoon view that we've blown up. Here's an example of the rough ER. You can see the little yellow dots where protein synthesis is actually going to occur on the ribosomes. We also have a smooth ER. And in the electron microscope here, you can see rough ER, the little bitty dots, and the smooth ER looks different, right, depending on if ribosomes are bound or not. The function of each, the smooth ER, has many metabolic processes, including the synthesis and the hydrolysis of different macromolecules. The enzymes of the smooth ER are mainly responsible for synthesizing membrane phospholipids. So where did we see this? Remember when we talked about lipids? Phospholipids, which make up that cell membrane, right? That double layer. This is where they're made. It's also responsible for the hydrolysis or breaking down of glycogen in the liver into glucose, so a very important step in creating energy. And then lastly, it detoxifies certain drugs and poisons such as alcohol and barbiturates, right? So this is where you can break down some of these drugs. Functions of the rough ER, they produce proteins for export out of the cell by packaging them into these transport vesicles for export. So look here, here's your rough ER, guys. There's my ribosomes. I've made some proteins. Once I make the protein, I have packaged it into these little vesicles, and you can see, where do they go? They're going to the next organelle, or the next stop in the endomembrane system, which is the Golgi apparatus. What type of cells would you expect to have lots of rough ER? Well, probably cells that like to make lots of proteins, right? Or have lots of proteins to make and eventually to secrete out of the cell. Our next stop along the endomembrane system, as we just saw, is the Golgi apparatus. The function is to finish, sort, ship cell products. We generally call this the shipping and receiving department of the cell. It's the center of manufacturing. We warehouse, we short, we ship. It's very extensive in cells that are specialized for secretion, right? So you can see this would represent a little vacuole that's coming from that endoplasmic reticulum. It actually fuses to the Golgi apparatus. And then these little purple dots right here, they move through the different layers of the Golgi apparatus. And as they move, they are modified, right? So think of it as like a shipping pack. The package comes in, then he needs to be packaged and wrapped and bubble wrapped and changed and labeled. And then eventually, he knows where to go and he's shipped off. Here's a nice electron microscope of that Golgi apparatus. You can see here's the receiving side. It's called the cis side. Vacuole binds. And then down here, which is the trans side, well, this is where the protein modification occurs. And then eventually these little vacuoles, which contain the fully modified proteins, will bud off and they will go wherever they're supposed to go, right? There's various places that they can go. The structure of the Golgi apparatus has these flat membrane sacs like this called the cisternae. It says it looks like a stack of pita bread. There are two sides. Again, the cis side is the receiving side, so they're receiving these vesicles from the endoplasmic reticulum. The vesicles fuse, they're modified, and then eventually the trans side is the side where the vesicle will bud off and then eventually go wherever he needs to go. So that's why we refer to him as the packaging and shipping department of the cell. This is kind of just the big picture of what I just said. I just wanted to show you, I referenced this in the other slide. So the vesicles come in, they fuse, they're modified, and then eventually they move off to where they're supposed to go. And here's again your cis side of your Golgi apparatus. And here's your trans side of your Golgi apparatus. Now, what types of cells, we'd referenced in a previous slide, so that normally cells that have a lot of Golgi apparatus like to secrete a lot of things, right? Because look down here, they're secreting off a lot of vesicles. What types of cells secrete? Well, mucous membrane cells, so saliva cells, for example, right? Think of things in your body that are constantly secreting something. The Golgi processing, so from the cis to the trans portion of this Golgi apparatus, the protein is tagged, it's sorted, as we said. The Golgis normally can also be referred to as the UPS center. And these little transport vesicles that eventually bud off are sometimes referred to as packages, right? They've been tagged with little barcodes, and they know where to go based on the barcodes that the Golgi apparatus has placed on them. So putting it all together, the big picture, remember our messenger RNA is going to come out here through the nuclear pore. On the rough ER, specifically on the ribosomes of the rough ER, proteins are going to be made. They'll be shipped out through vesicles. They'll fuse with the cis side of the Golgi. They'll be modified through the cisternae sacs. Then eventually, they will bud off as transport vesicles. And there's different things that this guy can do now. Maybe he's going to fuse with the plasma membrane and release his protein, right, for other cells. Or maybe he's going to go off on his own and do something else. We'll look at those possibilities. Our next stop along the endomembrane system is the lysosome. So the lysosome is a membrane-bound sac. It has these hydrolytic enzymes, right? So remember, lytic again means to lyse. So just like hydrolysis is to break a bond, hydrolytic probably means to break something down. The enzymes and membranes of the lysozymes are made by the rough ER and then eventually transferred to the Golgis. Again, remember from our previous slide, lysozymes are only found in animal cells, not in plant cells. And so these dark stain regions right here are some examples or where you can see the actual lysozyme in this electron microscope slide. The lysosome functions as the little stomach for the cell, so it's kind of responsible for, or it actually is responsible for digestion of macromolecules or old worn out cell parts that need to be recycled. Lyso means to break things apart, so means a body, right? And so they're also known as the cleanup crew of the cell. So they like to clean up old dead debris and parts. They'll break it down into forms that your body or the mitochondria can use in order to extract energy. The role in cellular digestion of the lysosome, you can see over here. Down here, the Golgi apparatus, again, has some vesicles that's come off. And so one of the roles here is that the lysosome can actually fuse with this food vacuole that the cell has taken in. Maybe this is like a really large starch molecule, right? And so the lysosome and the food vacuole actually fuse together, and the lysosome enzymes break down that molecule into smaller parts that now the cell can actually use. Another role here is the lysosome. You can see this is actually half of that kidney bean, half of a mitochondria. So it's probably just an old, worn out organelle within the cell that he doesn't need anymore. But the lysosome actually fuses with it and breaks it down into its molecular component, so the cell can also use that as energy. Of course, sometimes the lysozymes may not function or work properly due to a genetic mutation. But lysosome can't digest the enzyme or can't digest those particular lipids, particularly in the brain. We call this Tay-Sachs disease. So eventually what happens is the lysozymes begin to grow larger and larger within the brain. We call this a lysosome storage disease, and the lipids begin to build up within the brain cells. Typically, the child dies before the age of 5. So this would be an example of a lysosome function that has gone bad, right? It no longer works anymore, and it is a genetic disorder. You can't break down lipids within your brain. Of course, lysozymes are also responsible for some good things when they do work properly, when they're supposed to. So in fetal development, right guys, so the spaces between your fingers and your toes, as the fetus is developing within the womb, you can see here right now, the fingers are actually fused together, but it's the action of the lysosomes within the womb that will actually go in and break down the areas between the fingers. Of course, sometimes this isn't always perfect, so you can have fused fingers, and you may know someone that has fused toes, and this would actually be, for some reason, the lysosomes did not do their work right there, right? So lysosomes are good, they're necessary, we need them. They can also have negative effects when they don't work properly. And here you can see at 6 weeks, see how there's no little fingers there? He's really cute. And then at 15 weeks, you can actually see the spaces here between the fingers, and no ear, but you can actually see the definition here now. The ear has been carved out by the lysosomes. All right, our next organelle we're going to talk about is the mitochondria. So its function is to generate something called ATP. So ATP is the only form of energy that our body can use. So it would be analogous to, going to a foreign country that doesn't take a dollar bill. What do you have to do? You have to convert your money, right, into a form that that particular country will use, which is valid. So again, remember, energy, it isn't just automatically created, but when we take it in by the form of food, so maybe we're eating a really large meal, and there's lots of energy stored in those chemical bonds of that hamburger that you just ate. But how do we extract the energy out of that hamburger? Our body can't just use the energy that's released right away. So there's a very complicated process that occurs within this mitochondria that converts the energy that's in your food to a form that your body can use. And the only form that your body can use is this ATP form. Again, also the mitochondria, it's important to note it can only happen in the presence of oxygen, right, which is why we breathe in oxygen. It breaks down larger molecules into smaller ones to generate energy, also known as catabolism. And it generates energy in the presence of oxygen, known as aerobic respiration. So aerobic just means in the presence of oxygen. And this process of aerobic cellular respiration is a future chapter, of course. The structure of the mitochondria, you can see it actually has two membranes right here. It has an outer membrane, and it has a highly folded inner membrane. Do you see this? Right, so here's the outer membrane. Here's the highly folded inner membrane. This is called the cristae. And then there's also fluid here in between that inner membrane, right? This is called the mitochondrial matrix, so it's kind of analogous to the cytoplasm of the cell. It also has some DNA, some ribosomes, some enzymes. Down here it says, why do we have two membranes right here? So why is the inner membrane highly folded? Well, it increases the surface area, right? And as you increase surface area, you increase the amount of ATP made, because you see right here along this inner membrane, this is where ATP is made, at the location of the inner membrane. So the more inner membrane you have, the more ATP you're going to make, right? And this process, of course, is in future chapters. For now, just know that it has a highly folded increased surface area. It just helps you to make more energy. Here is my electron microscope, of course, which I showed you. Here's your outer membrane. Here's the cristae. Again, the cristae is that highly folded inner membrane, and then the matrix on the inside. Almost all eukaryotic cells have mitochondria, right? So there may be one very large mitochondria, or you could have hundreds to thousands of individual smaller mitochondria. The number of mitochondria generally is directly correlated with the metabolic activity, so we're also known as how active that cell is. The more activity, the more energy the cell needs, the more mitochondria the cell typically has. So types of cells that you would see with a lot of mitochondria would be very active cells, like muscle cells. You're using muscles all the time, right? Even when you're sleeping. Nerve cells, right? So you're constantly firing off signals between nerve cells in your brain and other parts of your body. So these types of cells tend to have very many mitochondria. I don't think that's right in English, tend to have large amounts of mitochondrial organelles. That's better. Our next organelle is the cytoskeleton. The function is support to maintain the shape of the cell, provides anchorage for organelles, so kind of a platform for the organelles to sit on within the cell. It also allows for movement, allows the cell to move, particularly the cilia and the flagella are two of these movement structures. And it allows for organization of structures and just activities within the cell itself. Down here in this picture, this is an actual cell. But this is a special type of microscope. It's called the fluorescent microscope. And you can actually tag certain molecules within a cell. So in this particular cell, they've actually tagged the cytoskeleton with a green fluorescent protein. So all this green is the actual cytoskeleton network within the cell. And they've tagged the DNA material with a blue fluorescent tag. So you can see the nucleus, and you're able to see the long, extensive network here of the cytoskeleton. Here are some examples of cytoskeleton components along with their functions. You don't need to memorize them, guys. I just wanted to give you some examples. Here are some actin filaments, which are responsible for structure, like plant structures. Some intermediate filaments, which example here would be the keratin, right? Protein that's found in your hair, makes it nice and strong. Tubulin subunits, which is responsible for the outer covering of this chameleon. So they have many different functions within the cell. The two particular extensions of the cytoskeleton I do want you to be familiar with are the cilia and the flagella. So again, these are both extensions of the cytoskeleton, so they just extend outside of the plasma membrane. The cilia are generally numerous and they're very short. Flagella tend to be one or two per cell. They're very long and they generally act in this whip-like motion. So they're both responsible for movement of the cell. The flagella is typically unicellular, they're usually very small, and it allows cells to kind of propel through water, so for movement. Cilia, which remember are very numerous, are responsible for sweeping mucus and debris from your lungs, right? So there's cilia that are all down your bronchial tube leading to your lungs. And another example is flagella is sperm cells, right? So sperm cells have that long flagella on the end, which helps it to swim. Cilia generally acts in this oar-like movement. You can see here's a beautiful electron microscope of a ciliate, and you can see the little extensions, the hair extensions out here. So it actually allows it to swim through this perpetuating forge axis. Flagella, on the other hand, usually uses this undulating movement, so it's kind of this back and forth whip-like movement, and this is an actual picture of a sperm cell. So here's the head, right, which contains the genetic information, and this is an example of a flagella, right, which obviously helps the sperm swim. All right, so your remaining questions, I didn't label them as practice questions. I forgot to redo this one, but I think there's a video here you can watch. Remember, you have to be in slideshow view. If you can't watch it, it's not a big deal. You should still be able to answer all of these questions based on what I just talked about, as well as your readings. There are many questions that you should answer. Here's your practice questions, right? I know it's not in, sorry, when you do it in slideshow, all of this won't show up. And this last question, which is number 31, as you can see right here, it reads, in a faraway city called Grant City, the main export and production, blah, blah, blah. This is actually going to be your challenge question for class. So it continues on this slide and this slide. And so these two slides, it has a little story, and it will be your challenge question to identify the components of the town and identify the analogous organelle, which has the same function within a eukaryotic cell. Okay, and I'll relabel that for class as well. And then here is an optional point opportunity if you'd like to, also known as extended learning opportunity. It's not required. But I will give you a few points. There's a biology and society question. A lot of it's opinionated. So, there's no right or wrong answer to just make sure that you actually explain why you have your opinion and make sure it's well thought out. Again, not required, it's just optional if you'd like to answer this question and upload it to the extended learning opportunity. So sometimes I may put in extra points for people who listen to the podcast. So here you go. Answer your questions, and on Wednesday we will answer them together in class.